My name is Morgan Swain. Welcome to Morgan's Chef Point, the modeling project checkpoint. In today's lesson of biology, we will be covering the stages of mitosis. I have created representations of the stages of mitosis using various ingredients. Here are some of the ingredients. The chromatin and chromosomes will be represented by red Twizzlers. Chromatin is made up of DNA, RNA, and protein. It is essentially the material which makes chromosomes. These chromosomes are carriers of genetic information. Sister chromatids are identical pieces of DNA which together form the chromosome. Two sister chromatids make up one chromosome. DNA, also known as deoxyribonucleic acid, is what holds this genetic information. In DNA, four nitrogenous bases attach to one another to form a double helix. Skittles will represent kinetochores. A kinetochore is the special structure located on the centromere of the chromosome where the spindle fibers attach. What is a centromere, you ask? Good question. A centromere is the location where two sister chromatids connect in the middle of a chromosome. The spindle fibers move the chromosomes by attaching to the centromere, which brings us to another ingredient. We will use spaghetti for our spindle fibers. Now, spindle fibers are the fibers which help move chromosomes as the cell goes through the division process. Spindle fibers are made up of chromosomal microtubules. Hence, these chromosomal microtubules are the building blocks of the spindle fibers. The nucleolus will be represented by a round buttermint. We will use dried papaya pieces for our centrioles and uncooked spaghetti pieces for our astral rays. Centrioles are part of the spindle apparatus. In order for cell division to take place, the centrioles must move to the opposite poles of the cells. Astral rays are thin fibrils, or essentially a cluster of microtubules, which help form the mitotic aster, along with other thin fibrils. Their job consists in making sure that the mitotic spindle apparatus remains in the correct position and orientation based on the polarity of the cell. And finally, we will create a cell membrane using blue frosting and a nuclear envelope using pink frosting. Before we transition into the stages of mitosis, let's give an overview of the cell cycle as a whole. The cell cycle is the combination of several phases which occur in order for growth, duplication, and division of the cell. The cell cycle includes the G1 phase, the S phase, the G2 phase, mitosis, and cytokinesis. In the G1 phase, all of the cell contents are duplicated except the chromosomes. In the S phase, also known as the synthesis phase, it includes the replication of DNA. In the G2 phase, the already duplicated chromosomes are checked to ensure that no errors have been made. If there are errors, necessary repairs are completed. Mitosis, also known as the M phase, is the essential process of cell division in somatic cells, which occurs after the G2 phase. Note that during mitosis, the somatic cell contents are duplicated. After the duplication, the somatic cell divides in order to form diploid cells, which are identical. The steps of mitosis, or the M phase, include interphase, early prophase, late prophase, metaphase, anaphase, and telophase. These steps prepare the cell for division. Finally, cytokinesis finishes the cell division by forming a cleavage furrow and splitting the individual cell into two distinct diploid cells. Now that we've covered several of the ingredients, let's see them in action. In this masterpiece, we see the first phase of mitosis, also known as interphase. During interphase, the chromosomes uncoil while other cellular functions continue. The cell nucleus is visible and bound by a nuclear envelope. The nucleolus is also visible in this phase, and the chromosomes have already been replicated. However, each chromosome cannot be individually distinguished. Hence, we see chromatin. Next, we have early prophase. In early prophase, chromatin has condensed, which allows chromosomes to be seen as two sister chromatids joined together by a centromere. The chromosomes, which have already been duplicated, appear thread-like. The nucleolus is disappearing. The centrioles are beginning to move to opposite poles of the cell. Fibers are starting to move across the cell to create the mitotic spindle. Now let's take a look at late prophase. In late prophase, the chromosomes shorten and thicken as they coil, and the nuclear envelope disappears. Kinetochores form on the chromosomes as proteins attach to each chromosome centromere. The chromosomes begin aligning with the help of microtubules attached to each kinetochore. Next, we have metaphase. In metaphase, the nuclear membrane can no longer be seen. The spindle has formed, and the chromosomes begin to line up along the equator of the spindle apparatus. This is known as the metaphase plate. Essentially, the metaphase plate is a longitudinal alignment of chromosomes at the equator of the cell. Spindle fibers assist in this process. Now let's cover anaphase. In anaphase, paired chromosomes separate from one another and move toward opposite poles of the cell with the help of microtubules. This process allows both poles of the cell to contain a complete set of chromosomes. Daughter chromosomes are what form when the sister chromatids of the original parent chromosome separate during anaphase. Daughter chromosomes contain identical genetic material of the original chromosome before mitosis had taken place. Now let's look at telophase. In telophase, the sister chromatids which began pulling apart at the centromere are now at opposite ends of the spindle apparatus. The nuclear envelope and nucleoli begin forming. Finally, we have the last part of the M phase, cytokinesis. Cytokinesis occurs when the cytoplasm splits the already duplicated cell contents into two cells. This pinching apart forms a cleavage furrow. The cleavage furrow only occurs during cytokinesis in animal cells. Note that the final product of cytokinesis is two identical cells, as shown here. Now that pretty much wraps up our recipe for mitosis. Thanks for watching and have a great day.